हॅलो फ्रेंड्स, वेलकम. तर या पार्टमध्ये आपण क्लासिफिकेशन ऑफ डी एन ए हा पार्ट बघणार आहोत. तर आपण स्ट्रक्चर ऑफ द डी एन ए मागील व्हिडिओमध्ये बघितला आहोत. अँड दिस इज ऑल्सो इम्पॉर्टंट वन, द क्लासिफिकेशन ऑफ द डी एन ए. विल सी हियर few more and start the classification of the DNA. Here the first, on the basis of the number of the base pairs per turn, the DNA molecule is classified into 4 types. The first one is a DNA molecule. This has pitch at of 2.8 angstrom and 11 base pairs per turn of the helix, and the depth of deep grooves is increased and that of the shallow groove is reduced, meaning there are 11 base pairs per turn of the helix. This is called as a DNA molecule. And the second, part 1, classification of the type 1, bDNA. And this is most common form of the DNA which found in the case of the eukaryotes. The bDNA is found in the eukaryotes, and this has a right-handed double helical structure. That is the bDNA. The pitch is 3.4 angstrom and there are 10 base pairs per turn of the helix. And the third one is cDNA. This form has pitch of 3.1 angstrom and 9.3 base pairs per turn of the helix. And the last one is ddNA. This is the rare form of the DNA and this has 8 base pairs per turn of the helix. And one more type, that is the zDNA. And we will see here this is the aDNA, and this is the bDNA, this is the cDNA, and this is the zDNA. These various types of the DNA molecule. This is one also A form of the DNA and B form, and this is the Z form. And this is one also A form, B form, C form, D form, and the Z form. We'll see the base pairs. On the basis of the number of base pairs, the DNA molecule are classified into A, B, C, D, and Z type. Move to the next, the location. On the basis of the location, the DNA molecule classified into 2 types. First one is chromosomal DNA. Mostly the DNA is found in the chromosomes. Such DNA is called as nuclear DNA in eukaryotes. These are the in eukaryotes. This DNA present in the eukaryotes, chromosomal DNA. The chromosomes are composed of DNA, histones, and RNA. The 3 components of the chromosome: DNA, one DNA, histones, and the RNA. Second one is cytoplasmic DNA. Some amount of the DNA is also found in the cytoplasm, especially in mitochondria and chloroplast. Especially exceptions are mitochondria, means no exception. The main cytoplasmic DNA present in a mitochondria and chloroplast, mainly present in mitochondria and chloroplast, and such DNA is called as cytoplasmic DNA. And this DNA plays an important role in cytoplasmic inheritance. This is one important. The cytoplasmic DNA plays an important role in cytoplasmic inheritance. And next point, the structure. On the basis of the structure, the DNA was classified into 2 types. First one is linear DNA, and the second one is circular DNA. Linear DNA, the DNA which is thread-like structure which with both ends free. In thread-like DNA means linear DNA is both ends are the free and is known as linear DNA. Such DNA is found in eukaryotes. The DNA is found in eukaryotes. And the second one is circular DNA. The DNA which is circular in nature, and the circular DNA is found in bacteria and viruses. The DNA of mitochondria and chloroplast is circular in shape, and during replication of a strand of the DNA molecule can replicate continuously and the other strand discontinuously. This is feature of the circular DNA, and a continuously replicating strand is known as leading strand, and while the discontinuously replicating strand is known as lagging strand. This is one important sentence. Example: the DNA of mitochondria and chloroplast is circular, and linear DNA present in the eukaryotes, mainly in eukaryotes. We'll see the pictures. We'll see this is the circular DNA, and this is the linear DNA, which is present in the eukaryotes. This is prokaryotic cell having a single circular chromosome attached to the cell membrane. Circular DNA, and this is the linear DNA. Eukaryotic cell. And stop here. We can, in next part we will see the replication of the DNA, which is most important part of this chapter. Replication of DNA. This is the dispersive three methods: dispersive, conservative, and semi-conservative. So thank you friends. We'll take a thank you.